Thanks for tuning in, everyone. You are listening to me, Todd Freeborn, and I am going to be presenting to you today this talk, Tissue Bioimpedance and its Collection, Processing, and Health Applications. So thanks for tuning in, and let's really dive in. So as we get started, you are listening to me. My name is Todd Freeborn. I am an associate professor in electrical and computer engineering at the University of Alabama in Tuscaloosa in the United States. And my research focuses on bioimpedance instrumentation, processing, especially using equivalent circuit models, and the applications of bioimpedance, muscle fatigue, muscle damage, soft tissue alterations. So that's going to form the core of what I'm chatting with you and presenting to you today. Before I get into the technical details and kind of weave this story about this area, I want to give recognition to the graduate students who've supported this effort over the last few years. So Bo, Shelby, Joseph, Tim, Nolan, Gabby, all have done great work in my lab and really helped advance the topics and helped generate the data and analyze the data that forms the core of what we're going to speak about today. So let's dive in. I keep saying this word bioimpedance, so let's start there. So as some background, the passive electrical properties of biological tissues exhibit the same electrical characteristics that the components we're used to working with. Resistors, capacitors, inductors all exhibit. So the idea is if we can measure these electrical properties of the tissue, we should be able to understand some of the underlying physiology or alterations of a tissue. And the interest of that is in assessing that bioimpedance alteration or current value for health-focused applications. Can we detect fluid shifts and changes? Can we detect muscle damage and injury? Can we detect blood pressure? Could we detect breathing? What is it about the changes in tissue that we can detect, and what information can we extract out of that? So while there are many health applications, my core focus is on skeletal muscle. So we're going to be chatting a lot today about muscle changes, alterations, and muscle is a complex tissue. So this just gives a high-level overview of the different features of how muscle is bundled and how we can look deeper into it all the way from a macro level down to a micro level and the filaments that actually lead to a contraction. And if we were to look under a microscope at these cells and these tissues, we would find that it is a complex interconnection of cells with fluids, intracellular, extracellular, different types of tissue, different geometries. So it's 3D in nature, it is complex, and it is not a simple electrical schematic in any way. But the tools we use to analyze circuits are still very helpful here. They still help us explore this topic. So at a very, very oversimplification, this technique of bioimpedance measures the voltage across a material. I've represented that as a resistor here, injects a current through it, measures the voltage across it, and then we can calculate that impedance as the voltage divided by the current. So theoretically, this high-level, pretty simple concept to grasp, but the details show just truly how complex it is. So when we're talking about this, while it seems very simple, it requires a lot. When we're doing bioimpedance measurements and their applications, we require instrumentation, signal processing, modeling. So all these skills that electrical engineers develop and kind of what our field is focused on is deeply involved in this. We all are probably very familiar with just how complex instrumentation is. The design of a current source, a controlled current source, is very challenging. The voltage measurement equipment is very complicated. So collecting all that information from a circuit perspective is still a very challenging problem. And now, instead of interfacing to conductors and wires, we are interfacing to a biological tissue. So we have the contributions of that interface. What does an electrode connecting to skin look like? What are the charge carriers? What impedance does that introduce? It's no longer a near perfect conductor that can be thought of as only a resistor. So what does that change? What do we need to accommodate in the design of that instrumentation? So my work builds on the amazing work that electrical engineers have been doing in the design of that instrumentation. So I am not designing that instrumentation, but I live in that area where I'm applying that instrumentation. I'm focused on the applications and how do we work between disciplines? How do electrical engineers and physiologists and exercise scientists and health researchers work to identify where bioimpedance can be applied and where it becomes a really useful health indicator. So beyond my own work, there are also other groups around the world doing amazing work in this field. They're exploring areas like tissue injury. I've bolded skeletal muscle fatigue because that's where much of my research is focused the last few years. But there is amazing work being done on the application of bioimpedance to identify and characterize neuromuscular disorders. So Seward Rutkove and Benjamin Sanchez are doing fantastic work in that area. We have the exploration of joint health, knee and ankle joint health, being done out of Georgia Tech and Dr. Einen's work. Excellent, fantastic work coming out of those areas. I recommend going and diving into their pieces if you're interested, learning more as you listen to this talk today. Okay, so that kind of paints some background of why we're interested, what we should care about exploring, but where does our work come in? So in the past few years, our team has investigated skeletal muscle fatigue, changes due to activity, implications of activity on those measurements, and the processing behind how you can clean data and start to ensure that you have high quality data for analysis. So that's a lot of where our focus is, but let's dive into that a little bit deeper. So at a high level, what we've seen is we've done a lot of work measuring the bioimpedance of biceps. So the bicep muscle is easy to isolate. So it's a great site of interest for exploring this work. So while the bicep is fatigued through activity, so think dumbbell bicep curls, lifting up a weight, curling it, putting it back down. So as we do multiple repetitions of that activity, we become fatigued. We are no longer able to execute that activity. And what happens to the tissue when that occurs? Obviously, something's changing. We can no longer do this activity that once a few minutes ago we're able to. So there are some changes here. So the question was, how do those changes alter the tissue bioimpedance, the electrical properties of the tissue? And can we use that? Could we explore that and use that in helpful ways? And I say helpful ways because fatigue is an important area to study. So why care about fatigue? Fatigue affects everyone across all age ranges, across daily living tasks, across work tasks. So fatigue and aging, as our skeletal muscle can no longer do the same amount of work and we get fatigued. Increases of fatigue can lead to increased fall risks. Falls have significant health impacts for aging adults and older populations. So it's important to maybe work towards how do we understand fatigue and its contribution, and could we monitor it, and could we make predictive tools about people's ability to do work in different areas and monitor that and modulate that so that we don't over fatigue and lead towards over injury. I say those words over injury because that also applies to maybe workers like nurses and construction workers who have very demanding physical jobs and have repetitive use injuries often, and muscle injuries often result of overuse or overexertion. So knowing that, how could we monitor and improve people's work conditions, what their timetabling looks like, their scheduling? Can we get in like peak performance while reducing injury that occurs over time? So another goal is to help reduce injury towards working populations. That has same goal is important for both athletes, tactical athletes, firefighters. We're working with soldiers and warfighters, and what can we do to help improve and reduce their risk of injury during training in those areas. So that kind of highlights fatigue as a site of interesting research for helping different populations. So what have we learned though? So the tissue bioimpedance actually shows a decrease as a result of fatigue. So if we see this graph here, we'll explore this in a little more detail as we go through this. But what also we notice is activity, so contraction, also causes an alteration, but an opposite one. It actually causes an increase in the tissue bioimpedance. So that gives a summary of some of the results we found. That oversimplifies it. So let's dig into the details. Let's explore what that means. So how do we learn this? What studies have we done, or what have we learned? So now we're looking at the experimentations with tissue bioimpedance. So what are we doing? What kinds of studies are we running? What equipment are we using to collect this information? So in some of our initial studies, we were interested in this question. So how does activity, specifically focusing on a bicep curl as that activity at different intensities, so 60% and 75% of the 1 repetition maximum, completed until exhaustion, alter localized tissue bioimpedance? So let's break down some of those pieces. I have these words, 60% and 75% 1 RM. That is your 1 repetition maximum. So that is an assessment of each individual for a weight that they could curl once successfully. So it's kind of normalizing across different participants. We don't want to use a fixed weight because participants have different strengths. They're able to lift different amounts. So this normalizes this activity for each individual participant. We talk about exhaustion. How did we reach exhaustion through this study? So we conducted repetitions until failure. So if we look at this figure over here, I have the number of repetitions for both the left and the right arm conducted through this study, and we see multiple sets. So we see at set 1, an average of almost 14 repetitions was conducted. Drop down to 9, and then drop down to around 6 or 7 at the third set. We see this conducting for 10 sets. So each set was repetitions until fatigue, until they were no longer to do another one. Between sets, each participant was given a 2-minute rest, and then they conducted the next set. So this took about 30 minutes per arm to kind of complete this entire set, but it resulted in near exhaustion. So by the 10th set, participants can only get an average of about 2 new repetitions. So they're exhausted. They aren't able to do this activity any further. So before and after, and also throughout this protocol, we collected the local tissue bioimpedance. So with AGCL electrodes placed on the bicep, as shown in this figure, we collected the tissue impedance across a range of frequencies using our Keysight E4990A, an impedance analyzer, a precision impedance analyzer. So what does this data say, though? So we have these measurements, we collected them throughout activity. What did we learn? What's going on when we look at this? So let's explore this figure on the left first. So we see we're presenting the resistance on the x-axis, the negative reactance on the y, and this is showing the change in resistance and reactance. So it doesn't provide any frequency information at this time, but the points on the right are that 10 kilohertz. The points on the left are 100 kilohertz. So we see some information about what frequencies we sweep from 10K to 100K, and we see this arcing behavior show up. So this is a very common trend in biological tissues, is this arc. We'll explore it a little bit further, but let's just look at the raw data for now. So the black line here is the pre-activity or pre-fatigue measurement, and then the gray line is that post-fatigue measurement. So we see a decrease in both resistance and reactance magnitude. So that's exhibited by a shift down for reactance and a shift to the left for the resistance. Okay, so pre-post change, an alteration, a decrease in resistance and reactance. Now, if we compare and look at a single discrete frequency, so if we look at 10K, 50K, and 100K for each set of measurements we've done, that's what we're presenting here in the right-hand figure. We have the mean resistance across all participants at each one of these sets. So we see a decreasing trend. So we see that decrease between each of the sets. Okay, so what does this kind of tell us? Activities leading to a decrease in the tissue resistance. Thinking physiologically about what this means as a tissue is exercised. there is an increase in the amount of blood flow to that region. There is actually sometimes an increase in the fluid based on tissue swelling, depending on how much activity there was. And if we increase the amount of fluid into a region of tissue, we've increased the number of charge carriers in that region. So unlike a resistor that we have at our benches, the charge carriers inside a biological tissue are molecules, charged molecules. So those molecules are the charge carriers. They reorientate based on an applied electric field. They move through that tissue. So when we have more charge carriers in that region, it looks like a lower resistance. So this lower resistance here, we believe is attributed to a fluid shift, a change in the fluid in that region of the body. So when we look at this though, right, so we conduct our analysis, we explore this feature. When we do our statistical analysis of this, we found that statistically significant decreases occurred at all frequencies for both groups at around the fifth set. So we saw small changes leading up to all these pieces, but statistically significant appeared at 5 and greater. So we do see alterations over time throughout this set. What's interesting though, so on the measurement of these participants 24 hours later, the tissue impedance had returned to its near baseline value. So it was very close to the pre-values, no statistically significant difference 24 hours later. So whatever alterations had occurred in the tissue as a result of that activity had resolved within 24 hours. So no non-reversible changes, likely no muscle tearing, no localized injury, like it was all likely attributed to swelling and fluid shift that resolve within 24 hours, which is also common and reflected in this type of protocol for other kinesiology studies. Okay, right, but we talked about what these decreases mean physiologically. What insight is this providing? If we can capture a resistance decrease, we can monitor localized fluid change. So how much fluid shift is occurring? Is that fluid shift related to the overall fatigue? How can we relate fluid shift to that fatigue? Questions we still need to answer, areas we still need to go towards. So the fact that it resolved within 24 hours leads us to believe there was no alteration of the underlying tissue. So we didn't induce any changes that are going to cause a healing factor or a recovery process that took significant time. So nothing related to muscle injury. But could these bioimpedance measures actually detect that? If we want to detect fatigue, if it's fatigue leading towards over-injury, we're going to need to be able to detect changes at the cellular membrane level, not just changes in fluid, changes in the tissue itself. So we need to change our protocol. So next we explored a eccentric activity protocol. So eccentric activity is when you are generating force but your muscle is being lengthened. So it's not enough force to counteract the activity you're trying to do. And while you're generating force and trying to contract that muscle and move something, it's actually being lengthened. So think you're lowering something very heavy that's heavier than you can normally lift. You can slowly lower it, but you could never lift it. Eccentric activity is very common in stair walking, downhill running, and for a bicep curl, it's lowering a weight beyond your 1 repetition maximum. So we're interested if that type of protocol induces a different time course of change in our biological tissue. The answer, we explored, we collected data from 5 participants, and this is a really interesting feature. So we exercised one arm, we did not exercise the other. So we have comparison data within each subject, and what we're showing here is 3 participants' data. The black lines are the impedance, again presented in that same way. We see that arc that's common that I was discussing previously. And we see that the black lines have some variation, but they're grouped tightly together. Variations expected, changes in daily living. So these measurements were collected before the exercise stimulus at 48 hours and 96 hours later. So they're all very similar for the unexercised arm. But comparing the exercised arm that did the eccentric protocol, we see a decrease in the magnitude of both reactance and resistance. That's similar to what we observed in the previous protocol, but at later significant time points. So the other protocol had resolved within 24 hours. Well, this protocol, the eccentric one, doesn't even show differences until 48 and 96 hours later, much later in the time course. That is starting to hint that these changes are taking time, hinting that towards muscle damage that is recovering and healing throughout this process. So what else do we collect through this to assess that and understand what was happening? We also collected measurements of circumference of the bicep and also of the participant's pain of that exercise bicep. Analog scale rated 1 to 10. How much does your bicep hurt when they came in for their additional measurements? So we collected measurements pre and post. So right before the activity, immediately after the eccentric activity, 24 hours, 48, 72, and 96. So immediately post and at 24 hours, we see no statistically significant change in this group. 48 hours, we're starting to see the decrease in the median resistance, but nothing statistically significant. But at 72 and 96 hours after that activity, we see a statistically significant difference in the resistance. Also observed in the reactance, another magnitude decrease. And this was true for 3 frequencies that were discretely assessed. 10 kilohertz, 50 kilohertz, 100 kilohertz, looking at that same range as we did with our first study on activity and fatigue. Okay, what's interesting about this? Where does this lead into? So in comparison to the circumference measurements, we measured the largest circumference of the participants, seeing an increase from their baseline measures at 72 and 96 hours. So swelling is occurring there. That's what we attributed that early data to, but now we're actually seeing that. We measured it. We captured an increase in that bicep circumference. Also overlapping some of that period is the largest change in report of pain in the bicep, right? So we see that swelling kind of induced delayed onset muscle soreness. There's soreness in that region. There's stiffness. There's swelling. So it supports that first, our eccentric protocol did induce changes in that localized bicep tissue. And then there's a recovery process as it heals from those, that eccentric exercise-induced damage. But the changes in bioimpedance indicate that this measure is also sensitive to those changes. So this is the first indication that yes, we can capture that potentially tissue damage and markers moving towards over-injury or damage. So this is exciting. This is a great and exciting feature that we want to chase. But it kind of leads to this bigger question. For both protocols, the change was a decrease in resistance. So when we're looking at these discrete single frequencies, decreased resistance was what was observed for both eccentric and the concentric protocol. So that's challenging. It's the exact same change, but at different times. So that's a hard indicator to pull out. So are there different markers that we can use? What other pieces could we use to differentiate the mechanism of change here? And this is where we get into more of the traditional and classical circuit modeling approaches. So a very common circuit model to use is the Cole impedance model. So this is a 3-circuit model parameter, 2 resistors, one looks like a capacitor, but I'm going to call it a fractional order element or a constant phase element. So it's a capacitor, but the J omega term represented as S here is raised to an exponent alpha, and we'll chat about that in a second. So this circuit model exhibits the same Cole arc, so the same arc that we see in that experimental data when we plotted it on a similar graph, but it has this impedance that's represented here, which we can split into the resistance and reactance components. What's interesting is this fractional order causes a decrease of that arc height. So really impacts the reactance component. We kind of see the impact of that in this expression of the impedance. This exponent was initially introduced when they were fitting early data sets as a fitting factor to fit the arc of this. But the implications of that is actually that this goes into the domain of fractional order circuit modeling. So this physically means that this model has a fractional order capacitor. This fractional order capacitor has voltage and current relationship that is dominated by a or described by a non-integer order derivative. So we could say that the current through this capacitor, this fractional order capacitor, is its pseudo-capacitance times the alpha order derivative of the voltage. So that has important implications for how you would model currents and voltages through this system. Here for the bioimpedance, we really just use it as a frequency-dependent fitting factor. But we do some really neat explorations of fractional order and fractional order calculus in some of our studies. So if you are interested in that, has piqued your curiosity, definitely go search fractional order circuits, fractional order calculus. It is a really, really interesting field. Okay, but I digress. Let's continue looking at the fitting aspects. So why do we need to use this? Can this circuit model help me understand and get a little more insight in the physiology of what's actually happening? Okay, so let's kind of back up a little bit. When we talk about this model, the resistances are often attributed to intracellular and extracellular fluid. So fluid flowing around the cells, total fluid flowing through the cells and around the cells. And then the capacitance, this fractional order capacitor, is linked to the cellular membrane features. Okay, so simplified case, this is resistors should be linked to fluid, capacitor, potentially the order should be linked to the cell membrane. So changes maybe in tissue structure, maybe tissue damage. So let's explore that. How do we explore this a little bit further? So let's apply this co-impedance model fitting to our bioimpedance data sets. So to do this fitting, what we're trying to do is we're trying to extract the Cole parameters, those two resistors, the capacitance, the order, determine which four of those parameters are needed to best fit our experimental data, which is shown here as that solid blue line, with the Cole model impedance expression. So we can use an optimization procedure to fit that. We use a least squares fitting, or I use a least squares fitting often to attack that problem. There are lots of ways to implement that, lots of objective functions. We're essentially trying to optimize a function and reduce the difference between that experimental data and this model with our specific parameters down to zero. We can't actually hit zero because nothing's perfect, but we're trying to get it very low, very small. And there's lots of different algorithms that you can use to do that search. We have least squares algorithms. We have particle swarm optimizations. We have meta-heuristic approaches. We have nature-inspired algorithms. So there's some really excellent work on the different algorithms that you can use to attack this problem. And which one is more accurate, or which one is going to converge to the global solution like faster and more accurately. But we're not going to focus on that area, but it is a rich area. So from a quick look and inspection, I've applied that least squares optimization here. We have a dotted line, which is representing the simulation using the extracted parameters to fit this experimental data set. We see great overlap between the area and the frequency band that's being fit, 10 kilohertz to 100 kilohertz. And then the rest of this dotted line is just the prediction of like what that behavior would be to much higher frequencies, down to the hertz and up into the megahertz. Because this kind of shows and links to the physical nature of those resistor values. So where this curve intersects on the x-axis for the resistance is where the two resistance values in that Cole model are. So that becomes a useful indicator potentially for the fluid inside a tissue, because low and high frequency should be dominated exclusively by fluid, because the cellular membrane should act like a capacitor and should be shorted or open depending on that condition. So that's kind of the insight that we use to kind of interpret some of these physiological mechanisms to this model. Okay, how well does it fit though? So we have that fitting visually, it looked great. When we do a comparison for all of our participants in that 60% 1RM group for our initial concentric study, we see errors less than 1% for vast majority of our data, less than 2% for the reactant. So that model is a good fit for these individual data points. Very accurate, it's doing a good job. Okay, so let's go into this. Let's explore and compare these features. So these are all the participants that we included in this study. The important features here, let's go look at this table down here. So we saw statistically significant decreases in the resistance components, R infinity, R1. So comparing the pre and the post, we see this decrease of about 1 ohm for R infinity. We see a decrease of around 4 or 5 ohms for R1. It's around decreases of 5%, about 13.2%. Aligns with the discrete comparison we did earlier of just the 10 kilohertz and 100 kilohertz resistance. So nothing really new there, kind of confirms that this circuit model does capture those same features, and we can interpret the fluid pieces likely through the resistance values of the Cole model. Okay. We initially started this because we wanted to explore if the capacitance is linked to cell membrane damage. So no change in the capacitance, no change in the fractional order as a result of that concentric activity. Okay. Does not violate what we were expecting, right? Like we didn't see changes after 24 hours, so that kind of aligns with what we already saw. But the bigger question now is there a change in the eccentric study? Okay, we've got this tool, we've got this model, let's apply it now to that next data set. So we're looking at that data again now. This is the eccentric activity study. We're looking at 3 participants. Solid is the experimental, dash is the Cole fitting. We're seeing some artifact in some of our data, which we'll talk about in a little bit. But again, we see a really good fit through that band of 10 kilohertz to 100 kilohertz. So this looks good. The fitting looks appropriate. What about that analysis though? Okay, so here is the time point data. Here are the extracted parameters for all of our participants. Okay, so we're looking at the medians of these for the exercised and non-exercised arms. Let's focus. So we saw the most swelling occurring at 72 and 96 hours earlier. Okay, well, when we look into the analysis of this data, we see resistance changes occurring at 72 and 96 hours. Okay, that aligns with our previous analysis. But now we do actually see a statistically significant difference in the capacitance of the model fit to that data occurring at 72 and 96 hours, which is not observed in the unexercised arms. This is only observed in the eccentric activity. So this is our first indication that the model and what we expect to be cellular damage from the activity is being captured and reflected in this Cole impedance model parameter. So this could be that marker that we want to use to detect injury, movement towards injury, as a marker for fatigue, potential injury. What could we use to monitor tissues? So that's really exciting, but it still requires a lot of exploration. This is preliminary. These have been small studies, healthy young adults, very limited activity. So we need to explore this further. That needs a lot more effort to really truly understand and characterize. But it's a great starting point for what we're trying to do. Kind of raises some other questions as we were going through this. We noticed that the fractional order of localized tissue was in this range from 0.7 to 0.75. There's been discussion and there's all these questions. Is the fractional order linked to the type of tissue? What's it related to? Like, it's a fitting factor, but what is its physical interpretation? How do we interpret what that fractional order means? So we were able to do a secondary data analysis to explore and interrogate this question a little bit. So the question was kind of, is because we saw skeletal muscle and localized tissues having a value of 0.7 to 0.75 in our studies, is this consistent? Like, is the fractional order linked to the tissue type? And would measurements of skeletal muscle from different body regions and different lengths of body regions also have that same fractional order? So that's what we dove in, and we're able to do a secondary analysis on a data set collected from a body composition study from work by Michael Esco in our kinesiology department here at UA. So this study collected impedance measurements of the right arm segment, the right leg, and then from the right hand to foot, calling that a full body measurement. So our hypothesis and expectation was, if skeletal muscle is the dominant tissue type and the fractional order is linked to skeletal muscle, the Cole parameters extracted from these three body segments should fall within that 0.7 to 0.75 range. So we analyzed data from 161 participants. When we calculated it, we have some samples of how good the fitting is, again, highlighting that Cole arc. The simulations are a good fit visually, but what happens when we explore that full data set? Okay, the important one here. Differences between resistances, not unexpected. Differences between capacitances, sure, not unexpected. We're really focused on the order. So we see here the full body distribution was very tightly centered around 0.75. We are expecting all three, though, to overlap very similarly. We actually see a shift. So the right arm had lower values, the right leg had higher values of alpha. When we look at the medians, we see differences. And when we do a statistically significant, or when we do a statistical analysis, the alpha value for all three of these body segments was statistically significantly different. So we disproved our hypothesis. We were expecting all of them to be similar, but they were not, which hints to us that limb geometry really impacts the fractional order also. It's not just the tissue under measure. That geometry is really important, and just highlights that we really need to do further investigation to identify which features of a tissue are really tightly coupled or strongly coupled to these model parameters. And is it just tissue type? We just proved no. So how much is fluid involved? How much is geometry? Because those insights will truly help us interpret this modeling feature and understand what bioimpedance is telling us. Okay. So that gives a deep focus into some of the skeletal muscle areas that we're working on. But for researchers interested in getting into this, let's think about some equipment, right? So what's going on? What equipment's available? What are we doing? So you can collect bioimpedance with many commercial instruments. I use a Keysight impedance analyzer. I also use an SFB7 from Impedimed, but we have instruments from Zurich Instruments, Solartron. These are all really excellent precision instruments. So when we think about them, like how hard can doing bioimpedance research be, right? Put some electrodes on the body, complete a frequency sweep, analyze the tissue data, right? I want to say be careful. Not quite as simple as that. And let's look at some of the features we found exploring different equipment. So we did a study because we were interested in how well do measurements from different instruments agree. Specifically, we looked at the Impedimed SFB7 portable impedance analyzer with a Keysight impedance analyzer with a test fixture that went to alligator cables. So that'll be an important asterisk. So we collected measurements from multiple locations on a forearm. So we had multiple electrode locations, but we essentially collected the same locations with both instruments right after each other. And so what you're seeing here in this figure is the black line is the SFB7 measurement, and the red line is the Keysight measurement. Okay, so black line, SFB7, has that arc behavior we're expecting. Keysight has that arc behavior, but then has a hook at high frequency. So a change in what looks like the reactance, and could be the resistance also, but it's hard to notice in this plot. What's different here? The tissue did not change. The tissue is in the same state. The time is very similar. There may be some fluctuations, but not that significant, not that large of a change. So this hints that the instrument and the fixturing of the instrument and how it's connected all impact that data. And it's important to consider that, because if we don't think of the instrument and the setup as contributing to what we're looking at with the data, we could interpret this hook as being something with the tissue. And that can lead us to incorrect assumptions. Now we're looking for models that have a high reactance in how we represent that tissue, and that might not align with the physiology of what's going on, and may actually lead us in wrong directions when we're trying to explore what models to use and how to interpret them. So it's important to consider how we're collecting that data. What we've actually shown here also too is that hook is often attributed to a parallel capacitance with the tissue under study. So it could be contributed from the instrument, could be parallel paths to ground, from the instrument to localized ground, so leakage away of current from your instrument. And you can model that as a parallel capacitor. And what we kind of showed is if you do that, you can extract that capacitance value and then get similar agreement between both your instruments. So the red line here is the resistance that was collected by the Keysight analyzer. The green line is the model values that best fit, using another optimization procedure that best fit this model. So determining all of them and the CP value. And then we remove the CP and do a simulation of the Cole model without that parallel capacitance, and that's the blue line. And we show that it shows very good agreement with the actual tissue values using the SFB7 as kind of like the reference standard, because it didn't have that hook artifact. This is an interesting way to solve this problem and can help remove some of these hook artifacts which are prevalent in a lot of measurements, because we don't often have complete control of the environment when we're collecting measurements. It's hard to determine where those parallel leakage pathages are when we're connecting to the human body with large cabling and different instruments. Okay, so that kind of hints and gives an idea of, okay, there's portable equipment. I say portable, you can move it from research lab to clinical lab, but they're amazing for getting that clinical grade research data, but they're not easily portable, takes a lot of effort. They're pretty expensive. We don't often want to move them. They're very expensive. They are not wearable. And this limits the exploration of tissue bioimpedance. So we're limited to research labs, clinical places, tightly controlled sites, which is where most bioimpedance studies are going on. But if we think of what the applications that we're reaching towards are, we kept mentioning doing fatigue monitoring for nurses, construction workers, soldiers. That doesn't happen in a lab. So we're going to need to transition these methods to a free living environment that can collect that data, analyze that data, report that data in real time, free living, in useful manners for this technique to truly be adoptable for health applications outside like clinical assessments. So we do work on that front also. So our group has a wearable platform based on a bioimpedance sensing integrated circuit from Maxim, the Max 30001. We've developed our own wearable platform. It can collect data from multiple tissue sites. So we're collecting both tissue impedance. We're showing the platform here. It's PCB, it's design, microcontroller based system. It breaks out the excitation measurement signals, runs them through some muxes so we can monitor multiple body sites, do some onboard monitoring too to monitor, self-assess its own operation, make sure it's working during field use. And then we can monitor different tissue locations. So here we're showing the knee. We're monitoring both below the knee, across the knee for different applications. And so now with this instrument, we're starting to move towards free living monitoring. But let's explore what we lose with that. Like what happens when we go from a really tightly controlled situation to this free living situation? What do we have to deal with? What gets introduced? What do we have to factor in when we're analyzing data and collecting data? Okay, so using this platform, here is some data. I actually talk about this data in another presentation for the conference, so definitely go check it out if you're interested. But what we're showing here is we're monitoring two tissue sites. So electrode pair E2 above and below, tissue pair E3 across, and this is during treadmill walking. So we're measuring the 128 kilohertz resistance at 2 sites during 20 seconds of treadmill walking. We see alterations of this tissue impedance. So we see how it kind of alters. We see this fluctuation of a few ohms around the average of each. So we see that movement, and we're attributing this to contraction artifacts, changes that impedance as we're measuring it. So now during dynamic activity, we have the tissue changing through contraction, and we have those tissue alterations being captured in our measurement. So we may want to use that or we may not, but we do want to be able to identify when that's occurring so we can actually either clean it or use it in our analysis. So we're kind of showing this feature again, right? So we kind of showed that impact on walking. Let's talk about that impact on another exercise we've seen multiple cases of, and that's that bicep curl activity. So monitoring, again, both multi-frequency on the left and then continuous single frequency on the right. We collected multi-frequency before and after, and then continuous data during an activity. So we see that during these bicep curls, we see an increase in the tissue resistance. And we also see fluctuations and changes in the magnitude of the reactance. So it's changing both components. We see slightly larger alterations, max values for heavier weights. So we're comparing 10 and 15 pound weights. So some hints that the exercise intensity causes an alteration and a change of the overall impedance alteration also. And then we see the first indications of like that. This is not an exhaustive protocol, but we do start to see the slight decreases between pre and post as a result of some of the fatigue, changes in fluid that result from these activities. But now collected by a wearable platform, no longer using a precision analyzer. We're opening up where we could collect this information, but we're highlighting some of the challenges we're going to face. Contraction artifacts, movement artifacts, like what's going on through these cases. Okay, what other factors here impact what we're doing? So we have contraction artifacts, we have movement artifacts, but we no longer have this precision equipment anymore. So we have this integrated circuit, and we have limitations of the integrated circuit. So because I'm not a designer of integrated circuits, I'm a user of those pieces, I'm limited by what has been designed. So this system, while single frequencies with that max 30,001 can be measured at 64 samples a second, multi-frequency sweeps like we do for characterizing the frequency-dependent nature of tissues require settling time and configuration time. So with this IC, you have to configure it. It takes about 10 microseconds, but then when it starts reporting data, the sigma delta ADC requires about 200 milliseconds of settling time before the data reported would be considered accurate or a good representation of what's happening. So if we're going to measure multiple frequencies, in this case, if we measured 8, 1, 2, 3, 4, 5, 6, 7, 8, required 8 configurations and 8 settling times, took a frequency sweep, or it took a time of 3.2 seconds to get one frequency sweep. Okay, well, 3.2 seconds can have a lot of activity changes. That means that we might violate some assumptions that are occurring when I interpret data, and I have to think about that when it comes into my analysis. So let's think about this. How does activity, muscle contraction, and sampling limitations impact the quality of bioimpedance? So let's tie this all together. So if I'm doing a multi-frequency sweep, I'm assuming that the tissue is in the same state at the start and stop of the sweep. So that holds true for very short time periods. But if I start collecting data over multiple seconds, there's a potential where that isn't true. I could catch a period where somebody goes from sit to stand. I could catch a period where somebody is walking and isn't even catching one instant of the walking, climbing up stairs, all these pieces. So muscle contraction and activity violate this assumption, and it can introduce artifacts into our data. Why is that a problem? So if we're going to use the co-impedance model fitting to extract information about the tissue, a change of a single data point or multiple data points in a single sweep can really alter our fitting. So here I'm showing two types of artifacts. So we expect, based on the Cole model fitting, that the resistance is going to decrease with increasing frequency. So the circles show that behavior. All right, but we have in each of these sweeps a single X, where one of those data points violates that assumption. So this can significantly alter the optimization fit of this data for the Cole impedance model parameters. That could change whether I identify a tissue as fatigued, not fatigued, cell damage, no cell damage. So I need to be very careful about the selection of data for that and its trend analysis. But this can also help me identify which data should be excluded. So I can eliminate data that violates those trends before I do that Cole model fitting towards improving what I expect to be a representation of the tissue. As we think about that too, let's talk about other challenges that can happen. So this becomes one way to clean our data. Other challenges that can occur are during movement dynamic activity, what's happening to our mechanics and the connections of our system. So we have the knee, it is moving, the electrodes on the body are going to be moving. If those electrodes are integrated into a brace or a textile, they can be pulling and moving away and pulled away from the surface. So we have an opportunity to actually disconnect our electrodes, and then through movement and activity, we could also even break part of our system. So it's no longer under a very careful view by a clinician or a researcher in a lab. We're in free living territory now. So lots of changes that we're not expecting can occur. So how do we detect that? How do we remove that data from corrupting how we analyze it? So if we have a disconnect event, so think of an electrode with gel connecting to the different, like our surface of skin and injecting current through it, we need good connection of this electrode to the body so that we can actually get that quality data. If it gets disconnected, current won't be injected, voltage won't be measured, we're not going to get an accurate representation of the impedance of that tissue. So how could that manifest? So another experimental data set, we have what we consider good quality data to be the circles, and then we have an outlier in the kiloohms. All the tissue data we've measured for localized tissue tends to be less than about 150 ohms. Segmental data, less than 200 to 300. Full body, less than 500 to 1,000. This is a localized measurement. We should not see a measurement that's 1 kiloohm, especially among sweeps that occur. There is likely nothing that has changed that tissue to spike to 1 kiloohm in this instant, the few seconds that it took to occur. What could have happened? During activity, one of these electrodes could have been pulled away from the body, disrupting the measurement. So maybe we're seeing an open circuit occur here through that measurement. So we also need to remove artifacts like this because this is also going to influence our Cole impedance fitting. So gives us kind of two ways that we can start to post-process and clean data before we do deep analyses of these. The trend artifact removal, making sure resistance shows a decreasing pattern, removing artifacts that exceed thresholds that we expect. So we see that here applied to data collected from a participant, 2 sites on the knee across 7 days. So wearable system worn for 7 days, in that 9 to 10 hours per day. We see nice, clean, continuous kind of transitioning patterns. We do see jumps at different periods, but not unexpected based on fluid shifts due to changes in positioning, such as lying down to standing, sitting to standing, standing to sitting down. Quick changes in fluid shifts can occur at any of those body transitions. So we're capturing all of those details through the knee and the leg in that time. For this data, when we removed and did this cleaning process, we had less than 4% of the data removed that violated the frequency dependence and less than 1% due to the threshold outliers, where we said anything below 10 ohms and above 150 would have been an outlier. So we have a very clean data set to begin with, and I want to kind of urge a caution here. Not all data sets are going to look that clean. So this was already a particularly clean one, and we need to do further work to really understand what are the rates of these artifacts, how do we ensure we've removed them, how can we be confident they're actually artifacts in the data. So we still have to do a lot more work in that area to help us answer that question. Okay, so let's talk about some of the early designs we had that really show some of our messy data. So our early designs experimented with flexible printed circuits. Here's data collected from 3 participants over 7 days. We've put all those 7 days together in a single data set. It's just one frequency, 8 kilohertz. Participant 1 looks pretty consistent for all the days, but day 2, we see some fluctuations, alterations. But look at participant 2 and participant 3. High variability data throughout the entire period. Low variability data through day 1 and day 2. Day 3 looks like a break event because we hit kilo ohm level data for the rest of the 7 days while they're wearing it. So we see that when we used a flexible printed circuit design, we did not obtain clean data like we saw when we used better cabling systems. And it led us into this question of like, why? Because what's actually happening? We observed cracking and fatigue of the copper traces as a result of the dynamic activity. So how did we find that out? This is an interesting like sidebar as we work towards flexible designs for wearable systems. When we inspected optically these flexible printed circuits that we used to implement some of the cabling and electrodes in our design, we found cracks at the interface of traces and often the pads of exposed copper. So in each of these red boxes, we see that trace crack. They all occurred at the interface of the polyimide overlay and the exposed copper, which is kind of like a hint to maybe some future explorations of how you can design a flex circuit that reduces that exposure and prevents some of these cracking. So we observed this cracking. We couldn't say for sure that it was a result of the activity because we didn't have measurements before our participants wore it. So we had to replicate and see if we could replicate that cracking in lab. So our braces have dynamic activity, not just bending and folding, but twisting. So we did both types of twisting activity on our designs so that we could see if we replicated those cracking. So here we have before and after photos, and after these twists, and as early as like 5 to 10 twists, we can start to see that cracking behavior. And so if this cracking occurs and we lose electrical connectivity, of course, we're going to see that high variability data that we saw here, especially this one. This one makes sense to have a crack that then just never came back. But this high variability data shows some of the insidious nature of cracking. And what do I mean by that? So we are curious, why does it fluctuate? And is there good data in here? Could we still collect good data and report good data? So when we looked at our broken boards, we took cracked flex PCBs, connected them to a known load with our impedance analyzer, and measured this model before flexing, after flexing, and during a bend of the flexing. So we see big scales here. So after flexing dominates. So we see kilo ohm measurements because the cracks are broken, not a good electrical connectivity. We're not getting a good measurement of this tissue represented by an RC circuit with hundreds of ohms in that kind of 90 to 40 range, which we see here in the subset. Then when we bend this broken flex PCB, it will make an electrical connection. And when you're sweeping the measurement, you can actually get a period of time where you have connectivity and you get a quality data measurement. So this broken design is insidious because during movement, it can actually reconnect through that movement and that force and give you good data, which leads you on a chase of why am I getting poor data sometimes and not others, and has this huge reliability issue. So this led us to scrap using flex PCBs in any of our designs after this, but it's still an interesting area of study because our current designs could have improved usability if we can increase the flexible nature of them and get away from some of our rigid designs that have to get incorporated into the textile braces that we're using. Okay, so let me kind of wrap this up and close this down now. You've heard me go on about the exciting applications, the changes we've observed, where our research is going, the wearables we've made for it, the challenges we face with the wearables. But where do we go? what is the outlook for this field and where are we hoping to grow towards? So we've really focused on bioimpedance data as the source of collecting and extracting information. But as we explore this further, especially with the artifacts and the errors that get introduced, I think there's this huge need for multimodal data. How do we identify activity context? When's the movement occurring? What movement is it? What's the pattern? Are we sitting? Are we standing? Because all those features influence the data we have. And I think it's going to be very important to understand that context to get insight for what's actually going on. We need to improve this artifact identification and cleaning and processing. We've shown some first steps that our group has done, and we're working to improve that so we can really be confident in the data we are analyzing to look for changes and say what is truly representative of the tissue. And I say that for free living conditions with wearables, because we don't have that same challenge in the clinic or research lab where we can just get a new measurement after we correct those conditions. And then we see improvements for wearable designs. So while we're collecting this data to explore the applications and where the site of application for health is going to be most important, that's not going to help anyone if no one's going to use this wearable and this design. So our early feedback is from some of our participants who wear this brace is comfort is challenging, especially with those wet electrodes. So how do we integrate dry electrodes that are comfortable? What materials do we need to use? How can we start reaching out for stretchable and flexible design pieces that can improve both usability and it's how it feels on a participant? And then the really big reach that I think is really important is we have all this instrumentation. We are excellent. And when I say we, I mean the groups that are designing instrumentation are doing amazing jobs at high quality designs, accurate data. We can collect bioimpedance data, but we really need to work with interdisciplinary groups to connect what our bioimpedance data is saying about physiological features of the underlying tissue. We can collect this data as engineers, but until we convince the physiologists and the medical practitioners that it can support the insights they need to help different populations, it dies. It doesn't advance any further. So there's that really big push to work with physiologists, exercise scientists, rehabilitation groups to really connect the dots here from collection to application and interpretation. And then for our group, as we think of skeletal muscle, really understanding what exercise intensity, type of exercise, injury, disease, what that does to the skeletal muscle and what we're trying to monitor through it. So really exciting field. I think there is so much exciting work to be done here. I hope this has kind of illustrated a really interesting area for those of you who aren't involved directly with bioimpedance. And I just do a quick shout out that a lot of this work was supported by the National Institute of Health, specifically the National Institute on Aging. And then if you have any questions, please feel free to reach out to me. I'm happy to field questions and chat about this stuff. I love it. I can talk about it for a long time. And I'm interested in hearing other people's work in the same area because there is so much exciting work that's happening and great areas to collaborate. So with that, enjoy the rest of the conference, everyone. Thank you for taking your time today to listen to this chat. And I look forward to hopefully meeting all of you in the future, maybe at TSP 2022. With that, have a great day, everyone.